The body uses two proteins, insulin and glucagon, to store and produce glucose to regulate the sugar in the body. When you eat food, your body digests and breaks down the carbohydrates, forming glucose. This activates the cells in the pancreas that produce insulin. When glucose levels rise, insulin levels rise, and glucagon level decrease.
When we take a closer look at the pancreas, there are two types of cells, alpha cells and beta cells. Insulin is produced in the beta cells. In the nucleus of the beta cell, the gene for the insulin protein is transcribed, producing an mRNA strand. The mRNA travels to the cytoplasm to be translated into the protein preproinsulin. When the preproinsulin is inserted in the endoplasmic reticulum, the signal peptide is removed, forming proinsulin. Endopeptidases then excise the C-peptide, creating a mature insulin. The insulin and the C-peptide are packaged in the Golgi apparatus into secretory vesicles.
The insulin travels through the bloodstream until it reaches a fat cell where it binds to an insulin receptor. Phosphate groups are added to the insulin receptor through autophosphorylation. Many proteins are phosphorylated as the signal travels through the cell. The last protein to be phosphorylated moves towards the GLUT4 storage vesicle pool where it binds with Rab protein, allowing the storage vesicles to merge with the membrane and use GLUT4 proteins to transport glucose into the cell.
When blood glucose levels are low, the hormone glucagon is secreted by the alpha cells and the islets of Langerhans in the pancreas, stimulating the production of glucose in the liver. After being synthesized, glucagon attaches to the GPCR in order to initiate the signaling cascade. This activates the G protein, which replaces the GDP with GTP. The G protein activates adenylate cyclase, which makes cyclic AMPs from ATP, activating protein kinase A. The protein kinase A phosphorylates phosphofructokinase 2 and fructose bisphosphate 2. This produces low levels of fructose 2,6-bisphosphate, allosterically activating fructose bisphosphate 1, which promotes gluconeogenesis, and suppressing phosphofructokinase 1, which promotes glycolysis. This leads to an increase in glucose.
Diabetes type 1 is inherited and affects insulin production because the body's immune system erroneously attacks its own beta cells, which destroys insulin production.
In type 2, there's insulin resistance, reducing glucose intake.